what you can also see here is that if you look at the different positions in the tRNA, is that the chemical modifications are much more frequent in the weak codon pairs because they are easier to tune by chemical modifications. And if you talk to people who are into evolution, they'll tell you that this was the way by kind of splitting the weak codon boxes in two, nature could actually introduce new amino acids that were not present in the old or in the origins of the translation apparatus. And one thing which I would also like to point out here is that this also limits, for example, claims of people in the field of synthetic biology, because you sometimes have the notion that, okay, we could basically take all the possibilities. So we have 64 combinations that are possible. Let's just choose one as a stop codon, and that would allow us to introduce into a synthetic organism 63 different amino acids, which would be great because then we can do all kind of funny stuff with it. The problem is you cannot do this because these interactions up here are so strong that you would really need to also work on the modification side to overcome these very strong barriers. With this, I'm at the end of what I wanted to say and which is part of the exam. So tRNA biology ends here, as does your script. However, I'll take another about 45 minutes to speak about some research from my lab and how we what we've done on tRNA modification, quality, and translation. Now, the fact that you're all here tells me that you're probably able to read quite well. However, there are people who have difficulties reading, and for them it's actually very difficult to succeed. However, if they succeed, they can be often very successful because they have managed to overcome certain obstacles that for us aren't even considered. Now, what I'm going to talk about is what happens if ribosomes are in the same situation, that they cannot read well. Now, what does a ribosome need to do when it translates? On the one hand side, it needs to find a tRNA where the anticodon perfectly matches its codon. And at the same time, it already synthesizes a peptide. So while it's reading, the nascent peptide chain emerges from the exit tunnel. And as soon as it does so, co-translational chaperones start to interact with the nascent peptide chain in order to fold it into its final three-dimensional structure. However, this means that those two processes actually need to be tightly coordinated. So while the ribosome reads, it already needs to fold its nascent peptide, otherwise this thing may actually not work. And all this happens in a very complex pool of many different tRNAs that can be modified, unmodified, charged or uncharged, from which the ribosome needs to select the correct tRNA. And you might be surprised, but tRNAs, as far as I would say, are still kind of like the dark matter of translation. So we know they're there are many of them there, but it's really difficult to quantify them and to characterize them. And you could ask why. I mean, these are just RNAs. I mean, we know a lot about RNA, we know a lot about DNA in the cell, so why, I mean, can't we just sequence whatever is there? And that's actually a problem, because I told you that there are many positions of those tRNA that are modified. And either these modifications are silent, so you can basically use an RT reaction and then you can synthesize through the modification, but then we can actually not monitor whether the modification is there. but much more often, or in many cases, these modifications are actually interfering with the reverse transcription reaction. So we cannot see the tRNA because the RT enzyme will simply terminate at certain modifications and stop the reaction. So it's difficult to say whether it's actually, if we see a change of tRNA sequences in our reads, it's very difficult to say, is this now a change of the modification or is it a change of the tRNA abundance? And that's why we know surprisingly little about even basic questions of how tRNAs participate in translation and how does this work quantitatively are questions that we know surprisingly little about because the quantifications are difficult. Now, you know from last week that modifications can occur in different positions, and what is important is that many of those modifications have been linked to disease. So if you do not have specific modification, and the one I'll speak about mostly today are these wobble-uridine modifications. So if you have those not properly modified, very often you end up in humans with patients that suffer under severe microcephaly. So they have very small cortices, and you can imagine that they're actually not doing very well, and most of them die at a very young age. What is special about wobble-uridines, this down here, is that they are almost always modified in pretty much every species under the sun. So this is really a modification of the ones that I mentioned with the codon wheel, which allowed cells to introduce new modification, new amino acids. So it's one of the modifications that splits a group of 4 codons into 2 separate codons. So it's basically modulating a relatively weak interaction and making it, optimizing it. And how this modification is brought about is shown here. So on the one hand side, you have the so-called ERM1 pathway, which puts a sulfur in position 2 of the uridine. And then you have the so-called elongator complex, which puts an MCM5 group here, a methoxycarbonyl methyl group. And this is abbreviated MCM5, so the methoxycarbonyl methyl, then is an NH2, which would be the NCM5. And then if you have this MCM5 position and the S position, the sulfur here, this would be MCM5S2U. So you have 3 tRNAs that have this full modification. You have 2 tRNAs that just have MCM5 and an oxygen here. And then you have 6 which would have this NCM5. Now, I told you that this is always present in any species under the sun. The kind of disappointing news now is if you knock those out under normal growth condition, not much happens. So you can, this is a yeast spotting assay where you dilute yeast cells. So you start with a given number here, you dilute it 1 to 5, 1 to 5, 1 to 5, 1 to 5, and so on. And you can see that kind of they get more diluted and you see less. And that allows you to kind of quantitatively assess how well they are growing. And you can see that the mutant spotted here, double mutant, grows more or less as fast as the wild type under normal growth condition. I mean, the double mutant grows a little less, but even that one kind of does fairly